Yeah, thank you, sir. So, a very good evening to all eminent doctors. Thank you so much for joining us today's webinar. Our endeavor is to spread awareness about rabies, which is fatal but can be avoided once appropriately vaccinated. Doctor, this is initiate from Cadila, thus its innovation to serve the society and humankind. So, Doctor, today the topic of this webinar is novel 3-dose G protein vaccine, ultimate key of compliance. So, Doctor, dear doctors, rabies is a worldwide novel 3-dose anti-rabies vaccine with great pride. And we would like to inform you that, sir, rabies has successfully completed 1 year of launch. And with more than 40K animal bite victims being vaccinated, 100% compliance was recorded and no case of adverse was registered. With this, it is my honor to introduce to you the speaker of today's webinar, Dr. Satish Kankaria. He is MBBS general physician. And now I would like to request Dr. Satish sir to initiate the talk, and later we will have two question and answer sessions. And all the participants can drop their questions in the drop box, and we will read out and all the questions after the presentations. So now I hand over to the sessions to Dr. Satish Kankaria, sir. Sir, please. Thank you.
Thank you, sir. I am Dr. Satish Kankaria. Welcome to this webinar by arranged by the Cadila Pharmaceuticals company. The main aim of this series is to introduce the novel 3-dose G protein vaccines, which is the ultimate key for the compliance. The rabies, it is the most important emergencies in India as well as in worldwide, because up to 59,000 of the human beings are lost their life because of the rabies, and out of that, the 20,000 human deaths occurs per year in India, and this figure comes to up to 60% in our Asia, and globally it is up to 30%. And out of this, only 9% death occurs in the cities, but 91% of the deaths occurs in rural areas of the India. So that it is major health problems, and around every person lost their life within 9 minutes. Now in rabies, we must know the treatment of the rabies. We can divide these rabies into the three types or three headings. That is a local wound treatment, that is the first aid, then active immunization and passive immunization. Again, the patient, when comes to the doctors should divide the patients into the three categories. Category 1, category 2, category 3. This category 1, where the only contact with the animal, either wild animals or the domestic animals, is there. There is no injury, no scratches on the skin of the patient. Generally, we confirm the patient is in category 1. We can give primary treatment and leave the patient. But in category 2 and category 3, we must give the active immunization as well as the passive immunization, and the local wound treatment should be given to the patient. Here, the first in group 1 categories, there is no treatment no active or passive management required. But in group 2, category 2, we should give the active immunization to the patient as well as the passive immunization to the patient, in addition to the local wound treatment. So that in all these three cases, or category 2 and 3, first treat the local wound thoroughly, clean the wound with the tap waters, and up to the 15 minutes. After thoroughly cleaning the wound, again we dry the wound, and after apply the antiseptic solutions to the wound. And if wound is large enough, we suture. If suturing is required to the wound, then in such conditions, we first inject the RIG, that is rabies immunoglobulin, in the wound. Confirm that the sufficient volume of the rabies immunoglobulins are infiltrated in the wound, which inactivate all the viruses which is located in the wound, and then we can suture the wound by loose suturing, and we should not suture sufficiently to the wound, and we should not give dressing to the wound. And then we have to give the passive active immunization. We start active immunization on the same day. As conventional dosage regimes, we are using the 5 doses of the anti-rabies vaccines. It is active immunization. We are using the cell cultured vaccines, and which require the 5 doses to complete the immunization to the patient. First dose is given on the first contact of the when patient comes to you on the first day. Then after second dose after 3 days, third dose after this 7 days, and fourth dose after 14 days, and lastly the 28 days. This is the dose schedules of the conventional anti-rabies vaccines. In addition to this, particularly category 3rd, where the much more skin or area is affected, mucous membrane affected, or major injury occurs because of the animal bites. In such condition, the rabies immunoglobulin should be infiltrated in the wound. Generally, the 10 units of the rabies immunoglobulin should be infiltrated around the wound, and remaining amount can be injected intramuscularly. Now, this is a standard treatment for the patient suffering or post-exposure to the rabies, or post-exposure after the rabid animal bite, where we have to observe the animal for up to 10 days. On 10 days, if their animals develop any symptoms, signs and symptoms of the rabies, generally animal develop signs and symptoms of the rabies within 7 to 10 days, then it indicates that it may have transfer the viruses in patient, and we have to give complete course of the rabies treatment. And if there is animal is healthy, there is no any signs and symptoms of the rabies observed in animal, then we convert the post-exposure prophylaxis to the pre-exposure prophylaxis. Now, this is a long treatment, particularly in both these patients, pre-exposure as well as the post-exposure, and patient fails. Most of the patient fails to complete this treatment. We can judge the compliance of the anti-rabies vaccines. The patient compliant means where the patient can complete the vaccinations. Then all the doses, scheduled doses, that is the 5 doses of the vaccines patient taken, it is a better compliance. Then poor compliance or delayed compliance means patient takes vaccination, but not a given schedule. It takes vaccines within 60 days. Then it is a delayed compliance. And next is the dropout patient who does not take all the vaccines, or either takes single first or second vaccines and does not completed the given scheduled vaccines. Now, there are few examples of the lack of poor compliance of this patient and leading to the death of the patients, where the reason of the death may be due to the lack of administration of the rabies immunoglobulins or poor compliance to the vaccines. In first case, that is a 58 years female patient having dog bites. That dog was killed immediately, and she has taken 3 doses of the purified C. camera cell vaccines, that is Rabipur vaccines. Vaccines taken at the 0, that is on same day, then after third dose, after third day, and after 7 days. But she has not given rabies immunoglobulin. Then it clinically, the dog clinically it is categorized as the cat 3 category. A dog having bites at the right side of the nose. After 20 days, it develops the paresthesia. Then there is a fever, restlessness of 5 days, and then alteration of the sensorium since 1 day. Cerebrospinal fluids were tested, which shows the cells, that is monomers, 5 monomers, protein 126, and sugar 71 milligram per dl. That is a low. The patient shows hypoglycemia and negative staining. The diagnosis of that patient was the rabies encephalopathy. Confirmation is done by the rising serum and cerebrospinal fluid virus neutralizing antibodies. Very increased virus neutralizing antibodies are observed in the titer of the 1:8,192 and 1:2,048 respectively after 5 days of the previous samples. And MRI also shows the bilateral brainstem mild hyper intensities, that is without any restricted diffuse or constant enhancement on day 2nd, and patient dies after patient dies in because of the after 16 days. After 1 day she died because of the she has taken lama, living without medical advice. Second patient was 48 years male, also having the dog bites, street dog bites, and status of the dog was unknown. He has also taken 3 doses of the vaccines, of the unknown type of the vaccines, and there is no rabies immunoglobulin was given to the patient. After dog bite, patient is categorized into category 3, and bite was at the left mid calf. After since 15 days, he developed dysphagia and restlessness after since 3 days. Cerebrospinal fluid was not taken because of the lama. Patient has left hospitals, and he developed rabies encephalopathy. Confirmation test was also not done, and MRI was not done. Then supportive treatments, since according to the protocols, was not given completely because of the patient leaves the hospital and the death occurs after 2nd days after leaving the hospital. And same with the third patient. He was a 34 years male patient having the street dog bite. He has also given 3 vaccines, 3 unknown vaccines, but no rabies immunoglobulin was given to him. Events, chronology, clinical features are the category 3 at the right dorsum of the foot. Since 17 days, he developed dysphagia and restlessness. Since 3rd days, and dyspnea. Then cerebrospinal fluid shows monomorphous 100%, that is 60 cells, protein 84, sugar hypoglycemia shows a negative staining. Diagnosis again, rabies encephalopathies, and confirmation is done with the rabies RNA PCR test is positive from the cerebrospinal fluids. MRI was not done due to early death of the patient, and treatment is modified. Malwinki's protocol was from first days, and patient dies because of the he has taken leave without medical advices. In all these cases, the deaths are due to improper administration of the immunoglobulin or poor compliance to the vaccination. It is observed that patient does not remain stuck to the given treatment regimes and drop out before completing of the treatment regimes. Next. 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 The compliance of post-exposure prophylaxis among the animal bite patients. The hospital, these studies were done at the Safadarjung Hospital, New Delhi, and it is observed in 360 animal bite patients retrospectively. It is observed that 53% of the patients were non-compliant to the full course of the anti-rabies vaccines. Compliant patient is 48% out of the 360 patients which are studied in the hospital in New Delhi. 46 patients was non-compliant, that was not taken treatment regularly or in dropout before completion of the full treatment, and only 6% of the patients are delayed compliant, that is they have taken the treatment but not in given schedule. And this is because of the long schedule of the treatment. Patient cannot remain adhere to the long-term regimes. Next. Next studies was performed at the municipal corporation hospitals at Bangalore. Here, study is done for the compliance to anti-rabies vaccines in post-exposure prophylaxis. 215 animal bite patients are studied, and each one of scheduled for the 1 dose of rabies vaccines administered intramuscularly on the day of the 0, 3, 7, 14, and 28 days as the post-exposure prophylaxis. And almost also measures content for compliance to complete the course. It is observed that after the animal bites, all these patients has taken, that is 215 patients has taken vaccines on first day. Then 12% of the patients drop out in second dose, that is on the third day. Only 88% of the patient has taken the second dose. After on the seventh day, third dose, only 85% of the patient has taken the third dose, and 15% of the patient drop out. Again, on the fourth dose, that is on the 14 days, only 70% patient has taken the fourth dose, and 30 patients has not taken any dose. And for up to fifth dose on the 20th day, only 60%, that is 40% dropout patient as observed. That is, patient cannot complete the given course of the 5-dose regime of the anti-rabies vaccines. The compliance rate for intramuscular rabies vaccines was 60%. The constraints for compliance to complete course of the anti-rabies vaccines it is observed that the interference, either there is interference with the school timing, so that 11% patients left distance of the hospital. Again, next 11% patient cannot take complete course. Then cost incurred or the wages loss of the wages, and 18% forgot the given scheduled dose. So that the major constraint for compliance to complete ARV was loss of pages, that is 47%. These are the reasons patient cannot complete the course of these 5 days course of 5-dose course of the anti-rabies vaccines. Next. Now, evolution of the rabies vaccines. That is, development of the more and more superior vaccine is needed to avoid the poor compliance in the patients. And we require the ideal vaccines. There is no any ideal drug in clinical practice that would produce the given actions only and which is suitable to the patient and does not produce any adverse effects. To obtain such type of drug or vaccine, the evolution process is continuously going on. Same happens with the vaccines. Previously, in 1885, the live attenuated vaccines obtained from the spinal cord was used. And that type of the vaccines we have to give into 31 injections of increase to required to increase the immunogenicity. These are non-standardized and dried preparations of the vaccines. Then to overcome these adverse effects, in 1911, it is tried that the another vaccines to evaluate that which is the inactivated vaccine, that is some brain products are used. It is a minimal neural content liquid preparations and it requires 11 injections, each of the 0.5 ml. So that this is also the lengthy procedures for the complete post-exposure vaccinations or post-exposure treatment of the patient. To overcome this, the cell culture vaccines are used. And these are the inactivated vaccines. These are the standardized vaccines, but the non-compliance lead to evolution of the treatment regime from 21 doses to 5 doses and still 1. So these type of the vaccines we are using here, that is more evaluated vaccines of the 5-dose regimes, which is a dose regime is 0 day, then 3rd, 7th, 14th, and 28 days. These are the dose regimes. And still these, and it is observed that some patient forgot the given dose, given schedule of the vaccinations or drop out because of the other causes. So that to overcome again this, the new vaccines is evaluated by the Cadila Pharmaceuticals companies to meeting the unmet medical needs and introducing a novel vaccine with simpler and shorter regimes. That vaccine having only 3 dose regimes, that is, it is a novel 3-dose recombinant rabies G protein vaccines. These vaccines it is the first protein subunit vaccines. It is protein particles of the viral antigens is used here, and it is prepared by the VLP platform technology, which is developed by the Cadila pharmaceutical companies, and produce these vaccines virus-like particles, and that virus-like particles can stimulate the immune systems. That is the first vaccine with 3 doses, which is given, and yet these vaccines can be given intramuscularly on the day 0, then 3rd and 7th days. Because of this short regime, compliance improves. It is better compliance due to easy to flow regime and less visit to the hospitals. And it is a convenience. Also, doctors also, because of the ready to use and no reconstitution is needed, so that it is a better vaccine to overcome the poor compliance of the patients. it is VLP technology is utilized here to prepare these vaccines. From the native viruses, the G protein is difficult to stabilize. But the G protein isolations by using the VLP technology purified and stabilized by the Cadila pharmaceutical companies. And this is the virus-like particles. These are protein in nature, protein complex, and these are available as a nanoparticles. So that these VLPs are similar to the native virus particles, but that do not contain the viral genome. So that because of they have no viral genome, they cannot replicate and cannot multiply after inoculation of the virus vaccines. But can mimic the viral antigenicity without being the pathogenic, that they does not produce the rabies like symptoms, and these are harmless. We can easily administer this patient. They are capable of inducing a strong humoral as well as cellular immune response to the patient. So that VLP virus-like protein particles, which are used in these vaccines, acts as a vaccines and are safer to administer to the post-exposure prophylaxis to the patients. Now, these vaccines is studied as a clinical phase 1, phase 2, and phase 3 is also completed. In data which is obtained from the clinical phase 2 studies, it shows the sufficient improvement. It tested against the more conventional used Rabipur vaccines, 5-dose Rabipur vaccines, and shows the equal seroprotection in patient even with third dose, where the Rabipur or other conventional vaccines require 5 doses to increase the development of the antibodies in the patients, that is seroprotection, which can be obtained by 2-dose study of the these new vaccines. Next, in phase 3, it is for the safety and effectivity of the vaccines, safety and immunogenicity of the novel 3-dose recombinant nanoparticle rabies G protein vaccines. These are administered as a simulated post-exposure immunizations, randomized comparative controlled multi-center phase 3 clinical studies were done on volunteers, and it is found to be the safe and having better immunogenicity in the clinical trial. And in this study, to evaluate the immunogenicity and safety of the rabies G protein vaccines, these are administered as simulated post-exposure immunization in healthy volunteers. Total of 800 volunteers are selected. Out of that, 533 subjects are tested for rabies, and 260 subjects are tested for Rabipur vaccine, that is reference vaccines, anti-rabies vaccines. In all these, complete doses are given, that is in rabies, 3 doses are given rabies at 0, 3, and 7 days. And in Rabipur, the 5 doses are given at 0, 3, 7, 14, and 28 days regimes. And it is observed that the rabies develops equal support, equal protection to the rabies as compared to the Rabipur, that is a conventional or reference vaccines. The results of these studies is that these vaccines having better efficacy to the standard routine 5-dose regimes, seroprotections on the day of the 14. It is tested seroprotection on the day of 14 as well as the seroprotection on the day of the 42. It is observed that in both these 14 and 42 days, the equal seroprotection occurs with the therapies as compared to the reference vaccines. This is for the efficacy, that is having equal efficacy to the reference vaccines. Then study for the safety. When safety is tested, the therapies having better safe or less adverse effects as compared to the Rabipur or reference vaccines. In complete course of these vaccines, the only 9.9% adverse effects occurs as compared to the 17.2% adverse effects with the Rabipur. It is tested for the local adverse effects as well as the systemic adverse effects. Local adverse effects in localized pain, redness, and swelling. In all these cases, the therapies are having less adverse effects. Localized pain, that is 7.69%, patient shows localized pain at the site of injection as compared to the 13.56% in reference vaccines. Redness, there is less redness occurs, that is 2.81% as compared to the 3.37% with the reference. And swelling at the site of injection, only 0.75% cases shows the swelling as compared to the 1.87% reference cases. Then systemic adverse effects are also tested. Therapies with the reference vaccines, the therapies produce less systemic adverse effects. These adverse effects like the fever, ear pain, headache, urticaria, joint pain, and nausea. Fever, to only 0.38% of the patient shows post-vaccination fever as compared to the 1.50% patient produce fever with the reference vaccines. Ear pain, the reference vaccine does not produce any ear pain. Therapies produce 0.19% ear pain. Only 0.90% of the patient shows the ear pain. Then headache, for headache, 0.38% of the patient shows headache after vaccination as compared to the 0.75% with the reference vaccines. Urticaria, again urticaria, the therapies develop urticaria, 0.19% patients after the therapies vaccination as compared to there is no urticaria observed with the reference vaccines. Joint pain, there is no joint pain with the therapies, and 0.19% of the patient reference vaccines produce joint pain, as well as the nausea also. There is no nausea with the therapies where the 0.19% of the reference vaccine produce the nausea. So that the novel 3-dose has better safety profiles as compared to the reference vaccines. And equally effectivity is observed in 3-dose as compared to the 5-dose vaccines. So that better compliance will be there. So that the novel 3-dose was non-inferior to the reference vaccine as a seroprotective. It is less adverse effects compared to the reference vaccines, so that the novel 3-dose was found to be safe and immunogenic and was comparable to the 5-dose of the WHO pre-qualified vaccines. This vaccine is used in India and it is awarded as the Pharma Innovation of the Year by the Economic Times and the Golden Global Tigers Awards, Malaysia, for the Innovative Product of the Year. Thank you. Thank you, sir.
Yes, thank you so much, sir. We really thank to Dr. Satish Kankaria, sir, for the informative talk, and we truly appreciate your efforts. Last but not least, we would like to inform you that Travis has won two prestigious awards in innovation categories, ET
Yes.
India Pharma Award and Golden Globe Tiger Award. So thank you, sir. Thank you so much.
Welcome. Thanks.
Sir, please have any question in question box?
No question.
Sir, just to give me a minute, I will check the questions in question
Yes.
box. Sir, Dr. Ravi Patil from Devla, he is asking what is the difference between 3 dose and 5 dose?
3 dose and 5 days. The 3 dose is a chick camera cell culture vaccines. It requires to develop complete protection, that is for up to 5 doses, whereas the 3 dose vaccines, it is the G protein recombinant technology is used here. Here the only G proteins, that are nanoparticles of the virus is used. There is no genome is used in these vaccines, and that virus-like particles acts as the antigen, which helps in the development of the immunity in the patients. Whereas in 5 dose vaccines, there are chick embryo cell culture vaccines is used there, and which requires 5 doses. Both these vaccines are to be given intramuscularly.
Thank you, sir. Sir, another question is that the benefit of 3 dose over 5 dose.
What mean?
Benefit of 3 over 5 dose. As compare 5 dose में 3 dose के benefit, sir, he's asking.
Benefits of the 3 dose, because of the 3 dose are given, that is less as compared to the 5 doses. The compliance is of patient is better. In 5 dose, the many patients drop out. We cannot complete the complete course of the treatment regimes, and because of the dropout, the treatment remain incomplete, and not perfect immune protection will be there, and may suffer. There are chances of the poor development of the immunity in that patient, and may suffer the rabies disease, or may develop signs and symptoms of the rabies. This is point of the compliance, better compliance with the 3 dose over the 5 doses.
Sir, last question. MRP. MRP is 715 rupees.
Yes, yes. As their price is more as compared to the 5 dose, but we require only the 3 doses of these vaccines, and there are 5 doses as it is comparable.
Yes. Thank you, sir.
Welcome.
Thank you so much.